The specific immunity or the acquired immunity is the sophisticated defense system of the body, and there are two types. One is antibody-mediated immunity, and the other is cell-mediated immunity. Antibody-mediated immunity is also known as humoral immunity, and it mostly involves the function of B lymphocytes, which end up producing the antibodies. The cell-mediated immunity basically starts with the helper T cell and involves a whole bunch of other T cells, such as cytotoxic T cells and memory T cells. So antibody is mostly B and cell-mediated is mostly T lymphocytes. Let's imagine that a virus has entered the bloodstream. This is the antigen on top of the virus, and the goal is for the B cells to make antibodies that will exactly match this antigen. However, we have millions of different kinds of B cells, each with its own antibody. So to find the correct match between the antibody and the antigen can take many days. So let's imagine that we pick the red antibody as the chosen B cell and the antibody that correctly matches the antigen. This stage of antibody-mediated immunity is called antigen recognition, and it can take several days. By then, the virus is already multiplying and infecting the body. Even though the correct B cell and its antibody has been chosen, the antibodies cannot be made until permission is received from the helper T cell. So a different kind of B cell presents the antigen to the helper T cell. It kills the pathogen first and then presents it to the helper T cell, which produces a chemical called cytokine. Cytokines have the ability to stimulate the production of antibodies in the next few steps. This stage is called antigen presentation, and the cell is called an APC, or an antigen presenting cell. And the purpose is to get permission from the helper T cell. Now that the B cell has permission, the chosen B cell then multiplies. It forms a clone of itself. Once it forms a clone of itself, it will be ready to differentiate and make antibodies. So these are the chosen B cells, the ones that have the ability to produce the correct antibody matched to the antigen. And this stage is called clonal selection. Once many new B cells are made, the chosen B cells are made and they multiply, they now differentiate into two types of cells. One kind of cell is called the plasma cell. These are the pink cells that you see here, and they have the ability to actually make the antibody. What you see in blue is the rough ER, and if you remember, the rough ER make proteins that leave the cell. Antibodies are proteins that leave the cell, which is why you see the rough ER here. The other type of cell that is the memory B cell, and these cells have the antibodies attached to them so that they will be able to recognize the antigen again when there is a future infection. So these are memory B cells that will circulate in the body. And this stage is called differentiation, and this is stage 4. The last stage in antibody-mediated immunity is the production of antibodies. As you see, this is a plasma B cell, and with its rough ER, very functional, and it's producing antibodies. The Y-shaped red structures that you see here are the antibodies, and they have the ability to target the antigen so that they may be marked for destruction by other cells, by other white blood cells. Let's review how the antigen presenting cell works. The antigen presenting cell could be a macrophage, it could be a B lymphocyte, it could be a monocyte. Its job is to engulf a pathogen. So here is your antigen presenting cell, and here is the pathogen. So its job is to engulf the pathogen, take it inside, and destroy the pathogen. And the next step is to display the antigens of the pathogen on its outer cell membrane. So here is the antigen that it's displaying, therefore the name APC, or antigen presenting cell. So it presents the antigen to the helper T cell, the T lymphocyte, which in response releases a bunch of chemicals known as cytokines. Cytokines are chemicals that activate the entire immune system. Once the T helper cells have been alerted about the presence of an invading pathogen, they release cytokines, and these cytokines activate non-specific defenses. They make them even stronger. Non-specific defenses such as inflammation become stronger in the presence of the T helper cell. Also, the T helper cell activates the production of antibodies by B lymphocytes. Once the T helper cell gives permission, the plasma B cells are able to produce antibodies. The helper T cell also activates a third type of cell called the cytotoxic T cell. Cytotoxic T cell, once they have been alerted, they can attack and destroy cells. These are cells that are already infected by viruses, and they could also be cancer cells. So they both can be killed by the cytotoxic T cell, and they can only be done once the T helper cell has given permission.